We're going to transfer to healthcare and life sciences. And with that, I want to introduce Ian Foster. He's a distinguished fellow and director of the Data Science and Learning Division at Argonne National Laboratory. He's also a professor of computer science at the University of Chicago. He's well known for his work with Globus, which links thousands of labs worldwide, and for his work building task parallel programming systems and applications. With that, thank you very much, Ian.
Thank you. Hopefully this will work. We'll see. So it's a great pleasure to be here. I'm giving a talk, as I'll say at the end, on behalf of quite a large and stellar team at Argonne. I particularly want to acknowledge my colleague Arvind Ramanathan, who sadly can't be here, or happily can't be here, because he just had his second child, so he's very happily staying at home there. So what I want to do is to talk about digital twins from two perspectives. First of all, an example that sort of has a global scope, trying to use computers, as we explained, to study the evolution of the COVID virus. And then it's sort of at a much more local scope, looking at how we are using digital twins within research labs to do self-driving discovery. Let's see if I can make this work. Yep. So anyway, so first of all, COVID. COVID, of course, is, we are all quite familiar with the phenomenon. The actual details of what goes on and that we, turns out, can model quite effectively it relates to at least one mechanism by which the COVID virus attacks a human cell, which is via the spike protein on the virus binding to a particular receptor on a human cell, the ACE2 receptor. This is something that, in fact, we can model reasonably well, it turns out, on using our high-performance computing systems to the extent that we can understand what impact minor mutations on the spike protein, the blue thing here, might have on its ability to, on the ACE2 receptor, on its ability to bind or not bind to the spike protein on the virus. So there's some fascinating science going on, trying to understand what this means in practice. And so in some sense, what we are doing here is building a digital twin of COVID viruses. But what we're interested in doing at Argonne is going beyond modeling the behavior of a single viral variant and its interactions with human cells to understanding how the population of viral variants may evolve over time and what impact that may have on the spread of the virus. So we end up proceeding as follows. First of all, collecting large numbers of viral sequences, building foundation models trained on those many tens of millions of viral sequences, some COVID, some not, and to obtain basically a model of the COVID universe that's able to predict what future viruses may emerge, and then combine this with other computational elements to be able to determine whether a particular viral variant, either in the wild, detected in the wild, or produced by our foundation model, as shown here, is likely to become a variant of concern. And so you can see, I think, that this is basically the bits that I show here are form a digital twin of the broader world of viral activities. And work that's going on at the moment is seeking to link this digital twin with the real world by building machinery for collecting information on new viral genomes, as in one direction, and then being able to propose or postulate or report new viral variants that may become detection targets for people trying to monitor what will happen in the future. So that's I think, very exciting project. I won't say more about it now, but rather argue that I think this application we might view as being an instance of a whole new class of scientific computing systems that combine what we saw here, AI, for building the foundation models that are used to predict future variants. Certainly high-performance computing to study the potential outcomes of a particular variant. Generative models, the AI model forms a generative model. In this case, we're not using robotic labs, but we will in the examples I show next. And other elements of machine learning and AI used to accelerate this discovery process. And we at Argonne, and I know several others here in the room, are increasingly seeing science through this process of combination of AI for, as shown here, filtering candidates, predicting properties of things in a very rapid, approximate manner, combining with simulations and with experiments, only when AI models are suspected to be insufficiently accurate or moving into spaces where new data are needed. So, very different view of how science may proceed, both in terms of these AI approximations to things and in terms of the use of AI in the loop to replace human decision-making as much as possible. Interesting implications for high-performance computing in this context. The role of HPC changes. First of all, we're using it a lot to train machine learning models. We're increasingly using it, though, also to perform simulations that have been defined for us by a broader AI-driven system. That simulation might, for example, be to find out whether a particular viral variant is likely to be able to attack the human cell and how its evolution might proceed over time. So it changes, of course, very much how we think about execution workflows. Another thing that interests me in particular is that, as shown in the previous slide and also reported in the previous talk, increasingly we ended up wanting to link scientific instruments or observatories, field observatories in the case of COVID, with high-performance computing, high-performance storage, etc. And therefore we need an automation ecosystem that can allow that to happen rapidly and broadly. I won't say more about that right now, except to point out that we, like the people at General Atomics, make extensive use of Globus mechanisms for movement of data back and forth, access to computers, and so forth. Okay, so let's move on, and I wanted to drill down a bit on one of the elements here in the lower right here, the robotics lab element. So, we have this sort of obsession at Argonne with working out how to accelerate discovery. So having an exascale computer, of course, is exciting in that regard. But having an exascale computer really just makes it more important to be able to accelerate other elements of this loop here. Once we've generated candidates, we've evaluated them using high-performance computing, we often then want to perform experiments to determine whether they work in practice. And so let's see if this... Yep, this is an omniverse simulation, by the way. Well, the one on the left is. The one on the right is the lab that we've established for evaluating and developing some of our robotic control functionalities. You can see that they look pretty similar. The omniverse one is slightly less messy. I guess that's not surprising. Anyway, the interest in our work here in autonomous discovery at Argonne is to use AI and simulation to close the loop. And by that we mean getting from the point where we can propose an experiment to perform based on perhaps an AI model, obtain the results, interpret the results automatically, and then use the results to define the next experiment to perform without having to involve a human in that decision-making process. And we heard in the previous talks how important that is to do if one is going to move at a pace needed to make required discoveries. So we've actually got a number of these autonomous research labs now set up across Argonne. The one that I just showed you is in the upper right, is actually used for development and training of students and so forth. So it doesn't deal with dangerous chemicals, but there are other ones that deal with biological samples. That's the lower right there. Others, the two on the left, that deal with dangerous materials, and so therefore they are air-free and otherwise access controlled. But let me say give a few more examples of how we're using these autonomous labs in the loop to, in a sense, as digital twins of systems that we want to develop. So here's a first one, which has some similarities to the one I started with. The goal here is to design enzymes for producing biofuels, for example. They could also be for other purposes, as I'll explain in a minute. Using, again, generative models trained on, in this case, protein sequences, or also gene sequences, which we then use to predict proteins, in this case, malate dehydrogenase, which is important for biofuel design. What's wonderful is using these techniques, we can very quickly come up with new molecules, evaluate them computationally, and then for those that work well, or look promising, rather, we can then synthesize them and test them in the lab, and come up with enzyme designs that reduce, in this case, the energy associated with catalyzing a particular reaction, which can translate into improved performance. And then I'll just give one more example, and that is work we're doing on antimicrobial resistance. I think people are probably by now familiar with this problem, so I won't go into details on it. But a particularly promising approach to developing new drugs for use against antimicrobial resistant microbes is antimicrobial peptides, small peptide sequences that can attack in different ways than current drugs. And again, there's this hope of being able to use generative models to basically act as digital twins for the population of possible antimicrobial peptides, and then use HPC, AI, and robotic labs to evaluate and provide feedback to refine our digital twin of the antimicrobial peptide space. And this is a sort of a high-level picture of some of the machinery that we're building for that purpose. So peptides come in. We use a number of AI agents to, first of all, retrieve information about particular peptides. Other agents, a structure agent that goes out and uses a combination of large language models and computational techniques to determine the potential structure of a particular peptide protein and its peptide and its interrelationships with a target microbe. And then another agent will go out and call upon other sources of information to evaluate the output. And those that look particularly promising, we can then go on to either run through HPC simulations or run experiments. And here, as in several other areas of our work, we're using large language models to increasingly automate this process of generating these agents, generating the protocols that they execute, and so forth. So simplifying what is otherwise a quite complex task. So here's just to give you an example of how one of these works in details, using a technique that's called retrieval augmented generation, which actually seems to work quite well here. So we want to know as much as we can about a particular peptide and its potential mechanisms of action in a particular space. We can't afford to go and read all the literature ourselves, so instead we, first of all, retrieve abstracts from PubMed that reference that peptide, and then use our best available large language model, which in this case was ChatGPT, to propose hypotheses that relate to the potential mechanism of action involved there. So to do that well, we first of all want a model with deep expertise, and we're engaged in what's called the Aurora GPT project to build a model which will have more expertise about science than ChatGPT, we hope. We'll see how we do. We also need the ability to be able to make millions of such requests because we've got millions of candidate peptides. And for that reason, we're looking at how to use HPC systems for another purpose, which is to serve large language model requests at high speeds and with rapid response times. Okay, so that's all I was going to say here. So here's sort of some summary and takeaway points that I would hope you could derive from this talk. So first of all, probably you're all here because you believe this, but HPC workflows are becoming increasingly fascinating. For a long time, HPC was single program, multiple data, rather boring stuff, to be honest. But now, increasingly, we're linking HPC with the external world in the context of, for example, digital twins, where data feeds in, flows in as computation is running, where computations reach out to the outside world to query data sources, for example, or to run simulations to generate new data, or to define and run experiments to collect data. Our computing infrastructure clearly needs to evolve to fit, to satisfy and to work within those contexts. I talked about the need for a global automation infrastructure. I think that's one important part of what we need to support these things. We're finding these foundation models, third point here, to be of great importance. I think we're still just, we and many others, I should emphasize, are just sort of starting to feel our way as to what sorts of models are needed. So large language models trained on scientific text are clearly important. Models trained on genetic sequences, on protein sequences, on viral genomes, etc., are also important, and they seem to be able to do all sorts of fascinating things. Now, we've got this wonderful set of slides that Arvin put together acknowledging some of the many people that have helped us with the work here, so I'll just go through those quickly. We've worked closely with NVIDIA, which has certainly continues to be a wonderful collaboration. We're working closely with people at Cerebras as well on training the large language models, for example. We've got a wonderful group of students, staff, and postdocs, etc., at the University of Chicago and Argonne, and also other collaborators at other places. So there we go. Thank you very much.
Thanks so much, Ian.